want to resume with our next speaker, Dr. Angela Genge. Angela is from the Montreal Neurological Institute, where she heads up a clinical research interest. She's a neuromuscular neurologist.
This presentation is a little bit different, but it is really to share a couple of different learnings that we're in the midst of in ALS. It is an example of when you get really frustrated by biomarkers. This is what we've been doing now in the ALS field, really as a result, and Jesse shares this history with me, of many years of trying therapies, not having good biomarkers to help us make go, no-go decisions, and an entire field coming together. So coming together to try to improve our biomarker situation, really in order to get effective therapies through faster. So I'm going to present to you a couple of different things in here. One is Capture ALS, which is a Canadian initiative to do a deeply phenotype bioimaging, biorepository, and biomarker project that captures, hence the name, the resources of all the ALS clinics in Canada. And it is actually not the only program that is looking at biomarkers in ALS, but I'll deep dive into this one after I present to you what is a very dense slide, but sort of gives you the idea of how important the field of ALS believes this to be. So what's happening in ALS, once we've hit many, many, many failures and only a couple successes in therapeutics, is really a simultaneous acknowledgement that we have to do better. And by that, I mean the Americans, the Europeans, the Australians, the Canadians, we have all at the same, over the last couple of years, in fact, it goes back more like 6 or 8 years now, have come to the conclusion that in order to move therapeutics ahead, we need both biomarkers to use to determine whether we're hitting a target in the disease, target engagement, and whether the therapies are actually changing what we believe to be key readouts, biomarkers that would indicate in a longer study that the drug would be effective. Because what we'd like to be able to do is make a decision much earlier than the end of a phase 3 trial as to whether we should continue with that treatment or go on to another. And listening to what's an absolutely fascinating series of presentations this morning, this has sort of reinforced my belief that this ALS approach could be used in a number of different diseases in neuromuscular disease. So what we have currently is several initiatives. In Canada, prior to Capture, we had something called Kalznick, which is really focused on imaging data in patients with ALS. And the concept is that Kalznick is a longitudinal, well-funded series of cases that have been enrolled into what we call a non-interventional study, where we're doing getting a lot of clinical data, including cognitive testing, the neurologic exam, serious clinical information, and imaging studies on these patients. And in those patients who pass away, we also have an additional pathology section to Kalznick. So this is the basis from which Capture has grown. But it's not the only initiative like this. Target ALS, which is now, I think, at least 6 or 8 years in the making, also is focusing on biomarkers and, in fact, collecting biospecimens in various different ways in collaboration with NEALS and the New York Genome Consortium. Answer ALS, which is one of the most robust of the current biobanks, has finished capturing, I believe it's 1,000 different patients longitudinally, and their approach has been to develop iPS cell lines of patients with ALS, to use these iPS cell lines to both define appropriate biomarkers and potentially help with the development of new molecules. Create ALS is another network, again American, in which they are using, it's a different group of sites that are using particularly genetic forms of ALS and trying to develop a longitudinal biospecimen bank to be used for the development of therapeutics. I left out NEALS because NEALS Northeast ALS Consortium, which is by far the largest, most unifying group. It's based out of Harvard with Merit Sokovich, and now I believe the two co-chairs are, co-chair leaders are Rick Bedlack and Tim Miller. And they have a biorepository that has been heavily used to look at biomarkers in ALS. On the European side, there's a genetic initiative called MINE-E, which includes, I don't know, 50 different countries around the world, and it's specifically looking at new gene mutations in ALS. Ambrosia, which is based in the UK, has a lot of both genetic data and imaging data. Although imaging is not ticked here, they're heavily linked with an imaging consortium. So Ambrosia is doing similar work, collecting biofluids to look for biomarkers in ALS. So you can see that the landscape is a lot of people figuring out that groups need to come together, labs need to come together, but most importantly, clinicians need to come together to start developing these biobanks or biorepositories in addition to other data, here clinical and imaging being the two other big pieces, and developing an infrastructure so that scientists who have what I call great ideas have a resource in which to test their great ideas, whether it be based on imaging or fluid collections. So what we came up with in Canada, I mentioned Kalznick, which is the imaging consortium, which is led by Sanjay Kalra out in Alberta. That is really focused on MRI data. Okay, it's launched, it's been going now for a number of years. It's now into its Kalznick 2, continuing to collect this set of data. But once at one point, we looked at this and said, well, this is a fantastic network. We're capturing a lot of patients across the country. It has a little bit of an American presence because eventually Sanjay brought in Salt Lake City and Miami. But we were clearly missing the biofluids and the focus on biomarkers that is so important in the new drug development world in ALS. So that's where Capture was created. It was created recognizing the gap. And when we looked at each of the different consortiums that I mentioned 2 slides ago, we realized that there was a Canadian opportunity with this. And I'm going to go through what the plan is, but we see the Canadian opportunity being the ability to do much deeper phenotyping than some of the other initiatives. We do very detailed clinical, historical, and cognitive testing, and to link in a lot of information we now have on the genetics. We have a number of geneticists in the country who are prominent in ALS and sort of tie in some of that genetic work and create the biospecimen collection in the open science format, which has been an important new development here at the MNI, because of an initiative, I believe it's launched now 4 years ago, where the MNI became an open science institution. The MNI is the Montreal Neurological Institute. What open science does is allow a setup in which the MNI does not claim IP, that's my big thing, on any specimens that they share with scientists or industry. And it allows for a very different exchange of information where both data and tissue specimens can be shared with researchers throughout Canada and actually internationally. So Capture is set up like this. Apologize for some of the graphics, but I think you understand how these get created. So Capture starts by doing what we call the dry biosamples, which is the clinical data, the Kalznick data, and includes a lot of MRI and MRS imaging, and adds in the wet biosamples. That has been a big discussion, but serum, the ability to grow iPS cell lines, CSF, and autopsy material is all part of the wet biosamples. They're saved in local biobanks, but there's a central biorepository, which is something we call CBIG. It's here at the Neuro. We have a fairly serious IT platform called Loris that allows us to keep the de-identified informed consent process stored. We actually now, through our ethics board, have the ability to do a virtual consent. Both the clinical, the imaging, and the biofluids can be stored either locally here at the Neuro, or because of a GUID system that has been set up, they can be de-identified and stored at a local biobank. And it creates a resource for our scientists, our researchers, to then develop their hypotheses and their questions, whether it be imaging or in the biofluid sphere, and then really go on to test their hypothesis and validate what we hope to be effective biomarkers going forward. The other important partner in the Canadian initiative is actually certain industry partners who have come on board because they have molecules or assets in the preclinical space where they want to be able to test on either cell lines or sera, or particularly those two groups of specimens. They want to test their molecule on these platforms in order to know either whether their molecule should go forward, or whether, if they have a host of different assets, which is the best one to move forward. The open science does not prevent that. In fact, in some ways it facilitates that, because there's any IP component to the partnership with industry stays with industry. We are not looking for IP directly, and that's pretty unusual in academia. And I won't tell you all the backstory of that. So what we're collecting directly are the clinical measures, the neurological and cognitive testing. We have a speech testing initiative, and obviously the imaging. I think the one piece that now I would consider adding in specific sites is actually PET imaging, because there's a lot of activity in PET now in the neurodegenerative space. We have electrophysiology and home genome sequencing, and blood, urine, and CSF, all part of this protocol. It's going to launch at 4 sites, but the concept is that it will include every site in the country as soon as everything is up and running. We have included patient involvement. Both, there's a partner advisory council. There's a platform that will be used by the patients. It's going to be led by one of our other colleagues who is based in Edmonton. So as we've learned in the last 3 months of how effective we can be communicating without seeing people in person, it's really opened up these kind of initiatives to much more involvement by the non-core sites in terms of patient engagement and in terms of clinician engagement. So we're pretty excited about having this as part of a platform of this biorepository. We have had routinely patients who are not eligible for treatment-related clinical trials still very much want to be involved in these kind of initiatives. So this is in part being driven by the patient advocacy, personal advocacy groups. It is actually backed by ALS Canada. And at this point, we're looking at federal funding so that it can be as robust as possible. Clinical assessments, these may or may not be familiar to all of you, but we've actually drilled down to the level of which cognitive testing we're going to do, what electrophysiology, and very specific things in our neurological panel. The biomarker, the biological materials are actually, this is an important piece of information, I think, for everybody on this in this webinar. We're actually set up to collect not only what's written in Capture, but we're set up to collect muscle biopsy and peripheral nerve and saliva for a broader, the broader initiative that biobank of the MNI that we call See Bigger. So See Bigger is a institute-wide biobank. It has already spun off a Canadian Parkinson's network project like this called See Open. It has spun off a local Quebec partnership. It has integrated another ultra-rare disease called hereditary spastic paraparesis, and that's connected to See Bigger. And finally, it has a growing muscle and this is really in the neuromuscular space. It also has a growing muscle biopsy collection, which is linked with See Bigger. There are other things, primary brain tumors and MS are also parts of the See Bigger program. But for the ones that touch the Capture, all of these are listed here. And because we have the background for Capture of See Bigger, we have all the SOPs developed, all the GOP-like SOPs developed. This is not rocket science. As I said, any institution could be that core for a network like Capture in any disease, in my opinion. It's just something that needs to be translated into the disease that you're most interested in. Obviously, for this group is adult polyglucosan body disease, but any of your major sites could be part of a core network like this that would certainly facilitate drug development interest in the field. The ultimate goal relates directly to this webinar. The ultimate goal is to find biomarkers. Currently in ALS, the biomarkers that we use routinely are limited to now neurofilament assays in the CSF. I expect by the end of this calendar year, we're going to have serum assays of the neurofilaments using the Simoa technology. There's a huge amount of effort looking at biomarkers that are specific to particular genetic mutations, and some that are slightly less specific. So there are efforts looking at SOD1, superoxide dismutase 1 is one of the important genetic mutations in ALS, and the RAM peptides related to another genetic mutation, C9orf72. With the third big effort that I'm seeing very active right now, looking at TDP43 and its ability to become a biomarker. Open science, as I mentioned, is the basis for what we're able to do with capture. But what we really want to use capture for is to stimulate researchers both in Canada but internationally, both industry and academic, to come up with some better biomarker technology. Target ALS put out a recent call for biomarkers in ALS, and we're looking at using this platform in at least 2 of the proposals, both direct partnerships and collaborations. I won't go over the neurocognitive assessments, but just to say this is something that can be well managed and very, very much something that can add to all of what we're doing in the biomarker field in ALS. The specimens I mentioned, the secret to doing really effective biobanks in any disease, and most of this has been recognized with the deficits in some of the older biobanks, and that is to make sure you have as much information on every specimen that you have stored as possible. So the deep phenotyping is a really important part of this, and if you decide to really expand out this kind of initiative in adult polyglucosan body disease, the ability to really make sure you have as much data around every specimen as possible makes a big difference in its usefulness long term. The imaging is the part that I left to the end because I'm not sure the imaging we're doing is very different than the imaging that has been discussed this morning. I think there's a huge opportunity when you combine biospecimens with imaging. So if you have a group in your network that is big on imaging and you have another group that is very much involved in biospecimens, I think bringing those 2 groups together to develop a resource like this could be amazing.